Here. Well, welcome back to Biotechnology. This week, we're going to look at protein structure and analysis. And in lab, we're going to be actually isolating the protein from uh, E. coli, the piglo green fluorescent protein. So we'll learn a little bit of direct experience with that. So, what we're going to talk about in this chapter is how proteins are made, and we'll look briefly at the process of protein synthesis, and then we'll talk about the structure of proteins, which includes the levels of proteins structure and also categories of proteins in terms of what they do. And then we're going to look at the functions of proteins, the roles in the human body, and then also how we apply this in the biotechnology industry, and look at some of the important proteins that are produced to treat disease or solve other problems. And then we'll talk about the methods for protein production and isolation and use the GFP protein as an example of that. So, a colleague of mine who's a biotechnology instructor likes to say that DNA is flash, but protein is cash. So, what do we mean by that? It turns out there is commercial value in proteins, and we'll talk about that. So, we want to first talk about what proteins are, what they do in the cell, and also their role in potential, as I said, disease treatment or other values. How are they made? So, how are they made in the cell? And then, how do we use that process to then produce them in biotechnology? And then, what is their commercial value or use? So, I just briefly included a couple pictures here, which shows a sort of an overview of the process of protein synthesis, comparing bacteria to eukaryotic cells. And that's important because sometimes we're taking a gene that's isolated from a eukaryotic organism, and then we're transferring that into bacteria to produce that protein. And so, what are some of the issues that might arise if we do that kind of thing? So, first off, I want to answer the question, what are proteins? So, proteins are large molecules, meaning they're made up of hundreds or thousands of atoms. And if you break them up into individual components, they're made of amino acids. So, we call them polymers because they're made up of repeated units of these amino acids. And they are absolutely required for the structure, function, and regulation of all living cells. So basically, all of our cells are bags of proteins, and those proteins are made because the DNA code gives us the blueprint for how to make those proteins. So some of the functions include enzymes, because most of the enzymatic or most of the reactions in our cells are catalyzed by enzymes. So they're biological catalysts. They speed up the rate of chemical reactions in the cell. We also have defense molecules, so we make antibodies in response to infection, but other organisms produce toxins. So many bacteria and fungi produce toxins to defend themselves, and some animals produce venoms, and either these are part of predation or part of defense. So there's all kinds of categories of defense molecules. And then if you look at structural roles, so what makes up the structure of our body, our muscle cells are arrays of actin and myosin, and they're involved in muscle contraction. And then if you look at the support that makes up the architecture of many of our organs, we have collagen as one important protein that gives sort of support and shape to our cells. So, in the process of protein synthesis, we talked about the flow of information. We take the information in DNA, use that to make RNA. And then, if we have a messenger RNA, we use that then to make protein. So, the process of taking the information in DNA and using that to make RNA is called transcription. And then, the process of taking the messenger RNA and using that to make a protein is called translation. And what you can see in the diagram here, that bacteria, since they don't have organelles, we can take the information from DNA, use that to make an RNA molecule, and then immediately translate that to make a protein. So transcription and translation can occur at the same time in the cells because there's no compartments. If you look at a eukaryotic cell, transcription in RNA synthesis is taking place in the nucleus, and there's some steps involved in that process that don't occur in bacteria. And then we have to export the RNA to the cytoplasm, to the ribosome for translation. So we have compartmentalization where transcription occurs in the nucleus and translation occurs in the cytoplasm. So a quick look at transcription. Of course, we know DNA is a double-stranded molecule, and when we're transcribing that RNA, we're going to synthesize from one strand of the DNA and use that to make that RNA molecule. So if our if our total genome in a cell is made up of thousands or many thousands of genes, when we make an RNA, usually this just going to be the code for one gene or one polypeptide. And that's sort of the approximation we made, that one gene is roughly the code for one protein. And we'll see there's some exceptions to that, but that's roughly the way it works. So once we've activated that gene and we know we need to make that protein, we need to make the RNA first. And then once we have that RNA, whether it's in a bacterial cell or eukaryotic cell, it doesn't really matter. We then are going to translate that. In other words, we're going to make that protein that's encoded by that RNA. And so the RNA process, or the process of protein synthesis or translation, occurs with the help of the ribosome. The ribosome is kind of like this decoder structure that's reading through the sequence of nucleotides. It's going to break them up into groups of 3 called codons. And so we always have a start codon that's always AUG. And then we read groups of 3, and that'll tell us what amino acid to put in each place until we reach a stop codon. So we join the amino acids together with a peptide bond, and that peptide bond is actually formed on the ribosome. So I've added a couple animations in explanation videos from Bozeman Science and a couple others just to take you through this process. I think you've probably talked about it in Intro Bio or at some point, but just a quick review of how that process works. So if you think about it, the linear code in the DNA is converted to a linear code in the RNA. Of course, we always have directions on our molecules, so 5' end and 3' end. And that gives us then a direction or order, then a linear sequence of our protein. So that's one view of protein synthesis, is basically just translating that code. A little more mechanistic look is a diagram here where it shows the messenger RNA, the ribosome is reading down the strand, translating it, and you can see the protein growing out the top, this growing polypeptide chain or chain of amino acids then. So what's the genetic code? Well, it turns out that we read the code in groups of 3, and maybe you've heard about the story about how this code was determined. We know that we have 4 bases in DNA and 4 bases in RNA. And if you think of a coding system, I have 20 different amino acids. And so how can I use 4 bases to give me 20 different combinations? So I can't use a 1-letter code. I can't use a 2-letter code, because if I have 4 bases and different orders, I could have 4 times 4, I could have 16 combinations. But if I have a 3-letter code, I have 4 to the 3rd combination. So I have 64 different combinations I could make, and that's more than enough to code for 20 amino acids. So this is one chart. I kind of like this one because it looks really cool and artistic. You may have seen a code like this, a chart like this. This is from the genetics book that I use, but you may have seen this in other biology books. But this shows all the possible combinations of nucleotides. And you can see that most amino acids have at least 2, in some cases 4, or in a couple of cases even 6 different 3-letter codes or 3 different codons that can code for them. So there's what we call a redundancy in the code. And what that means is sometimes that 3rd base position really doesn't matter. It's really the first 2 letters of that particular codon that are most important. But one exception is methionine. There's just one codon that codes for the amino acid methionine. So methionine is always first in every protein. And then our stop codons are codons that don't correspond to any amino acids. So UAA, UAG, and UGA. And this, of course, is looking at the messenger RNA. So we'll go through a little worksheet if you haven't done something like this before, where you take a sequence and then you use that to predict from the DNA to the RNA to the protein sequence, and you can follow that through then. So again, DNA is cash, protein is cash. So what is the commercial value of proteins? Well, we know proteins can be enzymes. So one place that we use them is actually in beer brewing. When you take the malt, the grain contains, that we're malting barley and so forth, contains a lot of starch. In order for the yeast to be able to ferment sugar, we need to break down that starch into individual sugar molecules. So this can happen during the malting process, the sprouting process, but we actually speed this up by adding the enzyme amylase. And this is produced through recombinant DNA technology. So we actually add a recombinant protein into the early formation of the malt to produce the material, the grain mash that the yeast are actually going to ferment then into ethanol. There's also some very important enzymes that are used in treating stroke patients or conditions where there's blood clots that are formed in the brain or other parts of the body. And these are enzymes that have been derived from bacteria or actually derived from the human body, but then cloned. And so these are really important therapeutic agents. You may have heard in treating stroke, there's that golden hour where they try to treat you immediately with the clot busting drug to eliminate oxygen deprivation in the tissues. So that's been really important. So lots of different enzymes have been produced for biotechnology. We've heard quite a bit about antibodies treating COVID infections. So these Regeneron monoclonal antibodies, where do they come from? Well, again, it's produced through recombinant DNA. Initially, it starts with cells from a mouse or a donor person, but eventually they're just produced in culture. So these antibodies are produced in large quantities and it gives you passive immunity. So treating this particular virus infection, the SARS-CoV-2 infection is one example, but there's other antibodies for other kinds of diseases. We actually use toxins. So if you've had anybody that's having their facial wrinkles removed, they've used Botox, which is botulinum toxin. It's actually stopping the ability of those cells, the muscle cells, to contract. So you relax those muscles and you relax the facial wrinkles. And that may seem a little frivolous, but actually the probably more important application of Botox is in treating patients with cerebral palsy where they have muscle contractions. It's also used for some migraine sufferers where they don't respond to other drugs. It relieves the muscle contractions that are involved in the skull area and relieves that. And then we have lots of different vaccines that are made up of proteins that are produced from viruses or bacteria. And again, this is in tissue culture. So you can see there's many areas in both pharmaceuticals and other applications for these proteins. So let's look at the protein structure for a second here. Proteins are made of amino acids, and the basic structure of amino acid, I'll just draw this a little bit differently, is you have a central carbon, you have a carboxyl group, so I can draw this a slightly different way. So C double bond O and then OH, so that's my carboxyl group. Then I have an amine group, so that's an NH2 group. Then I have a single hydrogen, so that's shown right here. And then I have an R group. And so it turns out they have 20 different R groups, and that gives us then the 20 different amino acids. But that's your basic structure of the amino acid. So here's another view of that. So this central carbon is called the alpha carbon. Here's my carboxyl group, here's my amine group, and then my R group. So if we look at the different R groups, you can look at chemical characteristics and group all of the amino acids into a few basic groups. So we have what's known as the nonpolar, which is mostly just hydrocarbons, so carbons and hydrogens. And then we have polar, which means we have some kind of oxygen or nitrogen on it. So we're talking about electronegativity, something that you may have talked about in chemistry or will talk about in chemistry at some point. So polar uncharged and then polar charged, where they have a basic group or acidic group on them. So this is a chart. I took this from the Campbell biology book that some of you might have used. And just to look at these in a little more detail. So non-polar side chain, so hydrophobic. So the smallest R group is glycine. It just has a hydrogen. We can get a little bigger. Alanine has a methyl group, but you can see everybody has these same groups: carboxyl, amine group, and the hydrogen. So all those things shaded in purple are the same. And then we just get bigger and bigger kinds of side chains and different names for these things. A couple things to point out here. Methionine has a sulfur atom in it, and phenylalanine, this is a benzene ring with a methyl group. And then this is called an indole group. And then this group on proline is interesting because the side chain is bound both to the central carbon and to this amine group. So this is the only amino acid that has that particular shape to it. Then our polar side chains, so we have hydroxyl groups or sulfur atoms or more hydroxyl groups or amine groups. So these are the different uncharged, but polar because they have hydrogen or nitrogen on it. And then electrically charged or charged would be, they have a negatively charged group like a carboxyl group or a positively charged group, an amine group on it. So these are the acidic and these are the basic amino acids. So kind of roughly the way it works, if a group is hydrophobic in a protein, it'll tend to cause the protein to fold inward at that point because it wants to stay away from water, because typically all proteins are exposed to water. And then if we have a hydrophilic group, they're going to be attracted to water. And of course, if they have charge on them, they also can be attracted to water. So this is another grouping, another listing. So it shows the charged, positive or negative charged, and then the polar groups, and then these non-polar groups. So we won't go into super detail about all the chemical characteristics of the amino acids. At some point, you'll probably take further chemistry classes or maybe even a biochemistry class where you go into this in great detail. But I do want you to just roughly understand that the side chains or the R groups on the proteins are really what determine how the protein fold. So you have these very long chain of amino acids. Think about dropping it into water. What's going to want to get away from water? So kind of like being a little oily patch versus what's going to fold out and be exposed to water. So that will determine the shape of the protein. So in order to join the peptides together to make our polypeptide chain and eventually make our protein, we have to join the amino group of one to the carboxy group of the next. And this is called a condensation reaction or dehydration because we have water as the leaving group. So what we're really doing, if I'll just draw this real quickly here, is I've had my carboxyl group here. This is my central carbon. So here's my hydrogen. Here's R group number one. Here's my amine group. And then if I draw another amino acid, so here's my amine group here, and R group number two, and my hydrogen, and then another carboxyl group. And what I'm going to do is join this hydroxyl group, or take this hydroxyl group out, and I'm going this carbon to this nitrogen. So that's what they're trying to show right here, was without showing the carbon bonds, there would be a carbon here. This carbon is joined to this nitrogen. So this bond here is referred to as the peptide bond. So every time we join amino acids together, we get a peptide bond. So that's what I'm showing right here. So the next thing we'll look at then is when we join all of these amino acids together, we can describe the protein structure, and we'll try to look at some different examples of proteins and how they fold and interact. So we'll stop at this point and we'll continue on in the next segment.